Hi, welcome to the cell cycle.
Interphase is the first step in the cell cycle where the cell begins preparing for mitosis or cell division. Interphase has 3 steps, although there is another direction it could go, referring to G0. The first step of interphase is G1, also known as GAP1. In GAP1, cells will rapidly increase in size, produce RNA, and synthesize protein. Before the interphase can move on to the next step, a G1 checkpoint will be activated to ensure everything is ready for DNA synthesis. Cell cycle checkpoints act like surveillance mechanisms that verify whether the processes at each phase have been accurately completed before they progress into the next phase. This includes monitoring the order of the phases, the growth of the cells, and the proper replication of chromosomes. These need to be regulated or else they will cause mutations and issues with the system. An example would be something like cancer, where the cell just keeps dividing. Now, in order for the checkpoint to be activated, we need cell cycle regulators, cisplatin and cisplatin-dependent kinase, otherwise known as CDKs. To drive the cell cycle forward, a cisplatin has to activate target proteins with the help of CDK enzymes. CDK enzymes phosphorylate specific target proteins, and the attached phosphate group acts like a switch, making the protein active or inactive. Cisplatin is what regulates the CDK, turning it on or off, which in turn controls the progression of a cell throughout the cell cycle. After interphase, the cell can either move to the synthesis phase, S phase, or to the G0 phase. G0 phase is when the cell stops dividing and can leave the cell cycle. It's also known as a rest period. This rest period can be temporary or permanent. A more permanent resting period would be like when the cell has reached the end of development and no longer needs to divide. This usually occurs after mitosis.
As for going into G0 phase after G1, it is usually because the environment lacks the necessary nutrients to enter the cell. If G0 phase doesn't occur, we move on to S phase. In S phase, we face the synthesis of DNA, where DNA is replicated so that the cell has 2 sets of identical DNA. After we finish the replication of DNA, we move on to the G2 phase, where we see the continued growth of the cell until cell division. The cell continues to grow and produce new proteins to prepare itself to split, like proteins for spindle fibers. The chromosomes in the nucleus should each consist of 2 connected copies called sister chromatids, which are duplicate copies of a single chromosome, along with the duplicate copy of the centrosome, which is a cellular structure with centrioles within it that helps organize microtubules and DNA and helps provide structure to the cell. Near the end of G2, we see another cell cycle checkpoint that will determine if the cell can proceed into mitosis and divide. However, this could all be nothing if something were to disrupt the cell. Disruptions in the cell can have many different effects, however, the most significant result in cancer. If damage is not irreplaceable, the cell will most likely continue to divide through with error and can lead to bunches of cells due to not having a signal to stop replicating, which is cancer. Because the damaged cells can continue to divide and carry out their jobs, this could accumulate more and more genetic errors over time as well. However, if the damage is irreparable, the cell may undergo apoptosis or a programmed cell death. The checkpoints also do this when they check whether the cell can enter the next stages. The next stage is known as mitosis or M phase, where the mother cell splits into 2 genetically identical cells called the daughter cells. There are 4 main steps to mitosis: prophase, metaphase, anaphase, and telophase.
In prophase, we have 2 stages, early prophase and prometaphase. In early prophase, the cell breaks down and creates structures to set the stage for chromosome division. The cell begins condensing chromatins, complexes of DNA and proteins contained in the nucleus, into compacted, visible chromosomes that remain compacted for the rest of mitosis. This is so that they become easier to pull apart later on. The spindle fibers from the centrosomes also begin to form in this stage, its purpose being to organize and move the chromosomes during mitosis. The nucleolus will also disappear in this stage, a sign showing that the nucleus is getting ready to break down. In the next stage, prometaphase, the nucleus and the nuclear envelope will break down and release the chromosomes that were held within it. The spindle fibers capture and move the chromosomes around. Kinetochores are used during this stage to connect chromosomes from the centromere, a region in the chromosome where the binding occurs, to centrosomes, and spindle fibers in order to distribute the replicated DNA from mother cell to daughter cells. After prometaphase, prophase has concluded and we are free to move on to the next stage, metaphase. In metaphase, the spindle fibers have fully attached to the centromeres of each chromosome, and the chromosomes will begin aligning at the equator of the cell, also known as the metaphase plate, in a single column. Two kinetochores of each chromosome should be connected to microtubules from opposite spindle poles. Before proceeding, the cell will check that all of the chromosomes at the metaphase plate are correctly attached to the microtubules. This is known as the spindle checkpoint and will assist in making sure that the sister chromatids will split evenly to each daughter cell. After metaphase, we have anaphase, where the division really begins. The sister chromatids are pulled to opposite sides of the cell towards the centrosome and are now separated.
Identical chromosomes are pulled toward opposite ends of the cell, and now each chromatid has its own centromere and can be considered a separate chromosome. At the end of anaphase, the cell should have twice the number of chromosomes that it did at the beginning of the cell cycle. Following anaphase, we have telophase, where two new nuclear membranes form. Each of the two nuclei contain the same number of chromosomes and same genetic information as the parent cell. The spindle fibers are broken down, nuclear membranes and nucleolus will reappear, as well as preparations for new cell membranes begin facilitating. The chromosomes will begin to decondense and return to their stringy form, just like in the beginning of the cell cycle. Before we move to the last stage of the cell cycle, why do cells go through mitosis? Mitosis is important for the growth and repair of our body, as well as asexual reproduction. Mitosis produces new cells and replaces old cells that are lost or damaged, so our bodies are not facing decomposition. For organisms that are single-celled, it continues their existence entirely. Without mitosis, life would not be able to exist. And lastly, we have cytokinesis that begins at the end of mitosis, which is the division of cytoplasm between two daughter cells. Cytokinesis is not part of mitosis. During cytokinesis in animal cells, a cleavage furrow is formed, which is the partitioning of the contents in the cytoplasm between the two new cells. A contractile ring will deepen the cleavage furrow and a certain membrane insertion phase begins. During their phase, a cell membrane is formed and inserted between two newly forming cells.